I'm Shriya Rangarajan and I'm working in Dr. Khorana and Alexander's lab. My topic is the spontaneous evolution of a breast tumor cell line featuring aberrant AKT crosstalk. Let me introduce the HC11 cell line. It's a mammary epithelial cell line isolated from the biopsy mice at mid-pregnancy. Normal mammary cells are arranged like that. They have an outer layer of basal cells and an inner layer of luminal cells. The basal cells express the cytokeratin K5 and the luminal cells K8. These are differentiated cells. Why do we use a cell line? Is because primary cells are very difficult to culture. And why the HC11 cell line? Is because they have a lot of properties very similar to the normal mammary gland. They're also p53 mutant. Now I'm explaining the significance of the HC11 cell line. It was found that under conditions of stress, the HC11 cell line underwent a transformation, and when sorted by flow cytometry, they gave two variant populations. The EpCAM positive population, which we're going to call the P variant cells, when injected into mice, did not initiate any tumors. However, there was another population of cells, the EpCAM negative cells, which were found to initiate tumors when injected into mice. This makes the study of these T variant cells very important. These were pictures taken under a flow microscope. You can see that there is a significant difference between the P and T cells. The T cells are more spread out. This may be due to the loss of the cell adhesion molecule EpCAM. These are pictures taken under a confocal. The green cells are basal cells, while the red cells are luminal cells. Like you can see, the T cells do not show either of these cytokeratin markers. This shows that the T cells are undifferentiated cells. Now, what do we know so far? The T cells are tumorigenic, and they initiate tumors when injected into the mice. They are an adaptive cell type, and they are able to transform under environmental stresses. Therefore, it becomes very important to study the differences between these two cell types. We now treated the T and B cells with different growth factors. EGF is found to induce cell differentiation, insulin for cell proliferation. When we treated both T and P cells, we found that under all conditions, the P cells do express these cytokeratin markers. However, T cells do not show these markers under any conditions. We counted those cells in the P for the basal and luminal fractions, and we found that Wnt marginally increases basal cell population. However, this is not the focus of my study. I will now attempt to characterize the T cells. I'll introduce the PI3K and MEK pathways, which are pathways frequently indicated in breast cancer. They are found to be upregulated, and the effect of these pathways is an inhibition of apoptosis and increased cell proliferation. Insulin specifically activates the AKT pathway, and EGF activates this MEK pathway. Now, why do we study these pathways? Because it was found that when we treated the B and T cells with phospho-S6, this phospho-S6 is a downstream protein of the AKT pathway. It was found to be expressed only in the T cells. This suggests a dysregulation of the AKT pathway in T cells. So the question arises, are the signaling responses of the PI3K and the AKT pathway different in T cells? We use two methods. One is gene sequencing, and another is we test the responses of these cells to inhibitors of these pathways. This is the protocol I used for gene sequencing. We use genomic DNA, mRNA and cDNA. We use cDNA because we could cover larger sections of the genome. Then we ran it on a gel and sequenced if it ran. But more often than not, it did not amplify, and we were having problems with the sequencing. This is a strategy we use for sequencing Hras. We we concentrated on two genes. One is Hras and one is PI3K-AKT. Why these genes is because they are constitutively activated in breast cancers, and this leads to permanent activation of the pathways. Codons 12, 13, and 61 are frequently mutated in Hras, and we used these two primers and covered almost the entire gene. Like you can see, we have covered these codons, and we did not find any mutation. For PI3K, it's a much larger gene, and they are found to show hotspots of mutation in the exons 1, 2, 9, and 20. So in order to cover as much of the gene as possible, we designed 3 sets of primers to cover this section, and that. What did we find? We found that Hras does not have any mutation. Exon 9 of PI3K-AKT does not have any mutation in both P and T cells. These sequences, when I say they don't have any mutation, they are compared to the normal BALC sequence. And we could not complete the sequencing. Exon 20, we found multiple bands were forming on our gel. We don't really know why. We probably have to redesign the primers or use different conditions. I'm going to introduce the cell proliferation assay, also called the WIST1 assay, which I'm going to use in my future experiments. The principle is the reduction of tetrazole to formazines by viable cells, and we measure the absorbance at 440 nanometers. The protocol I use is I seed cells, I change the medium, I add WIST1 on the 3rd day, and I measure absorbance 4 hours after treatment. Now this is a very interesting result we got. We found that wind significantly increases the proliferation of T cells alone as compared to the P cells. Wind under normal conditions does not induce proliferation of any type of cell, mammary cell, epithelial cell, but it does affect T cells. This correlates with data by Soyoung, where she found that phospho-AKT was significantly increased in T cells as compared to the P cells under the influence of wind. And since wind is an activator of the AKT pathway, this again suggests a dysregulation of the pathway. So what did we do? We treated the pathway with 2 different inhibitors. The MEK pathway at this point with the U0126, and the PI3K pathway with LY294002. And we found that both cells seem to be equally susceptible to inhibition of the PI3K pathway. However, we found that when treated with an inhibitor of MEK, the T cells were less susceptible to inhibition of MEK. This is a previous study, which was found that when the HC11 cell line was treated as a whole, inhibition of the MEK pathway seemed to upregulate the PI3K pathway. And like you can see, there is an increase in phospho-AKT when treated with MEK. So this would be a future study. To summarize, the T variant cells, they suppress differentiated cell markers like K5 and K8. They divide at approximately the same rate as compared to the P cells. They express phospho S6, which is a marker for AKT. They show AKT stimulation in response to Wnt ligands. They are resistant to MEK inhibitors, and they do not contain any mutation for the Hras gene. That's all, thank you. I would like to thank the Khorana program, DPT and IDSSTF, Dr. Caroline Alexander for mentoring me, Soyoung and Rod Clark, all my lab mates, Dr. Ansari, and all of you guys. Thank you so much.
Sequenced Hras. Do you know how? Did you have a program that could recognize heterozygous mutations, or did you examine traces?
We compared it with the BALC